Hi, everyone. This is the second seminar from the CFSD food cluster. Our speaker today is Shaelyn Xu on the ecological dynamics of microbials in food processing facilities, pathways to improve sanitation. So Dr. Xu, please take over.
Okay, thank you, Philip. Thank you for the introduction and good morning. No, maybe good afternoon, everyone. Thank you for being here today. This is actually my very first ever talk as a freshly minted PhD. So I'm very grateful many of you are able to make it here. What I'm gonna do is I will share my PhD research about how the microbial dynamics in the pork processing facility to reveal what microbes are there and also to provide the information on how we use ozone nanobubble as a novel sanitizer to improve the sanitation efficacies. So as I said, in this talk, I will mainly focus on two. The first is to reveal what microbes are there, how they persist and disperse across a processing facility or over time. The second one is I will show my findings of how using the ozone nanobubble to control the microbial spoilage in both based on the lab result and the pilot scale result. Ultimately, I want to use those two findings and then provide insights for the improved antimicrobial interventions. So let's start with a fundamental question: how microbes are introduced to the food? Food processing facility typically operates as an open system where they have constantly intake microbes from raw materials, from the water, air, as well as soil. The contamination of microbes to food can be either come from the raw materials or from the food processing facilities. For example, some plant endophytes are naturally colonized on the plant tissues and then can be transferred over to the food products. Some human-related bacteria, such as Staphylococcus aureus, can be introduced by the improper human handling and then further contaminate the food products. The microbes resident in the raw materials can be also introduced to the food processing facility, and then they start to colonize on the equipment surfaces and then stay as a resident microbiota in the processing facility. Those resident microbiota serves as a subject to change. This is mainly because there are some abiotic factors and biotic factors influence their dynamic and diversity of the microbes. For example, the sanitation protocol as well as the operation temperature certainly will inhibit or favor the growth of certain microbes. And then the microbes can also interact with each other or form biofilms as the biotic factors. And then the formation of biofilms usually can lead to their long-term persistence. So a bit introduction about biofilms. Biofilms are complex microbial ecosystems formed by one or more species in an immersed extracellular matrix, which is mainly composed of polysaccharides, proteins, DNAs, and lipids. This extracellular matrix actually works as a physical barrier that limit the diffusion of the biocides or sanitizers reaching to the interior layer for microbes, thus impedes the sanitation efficacies. It has been considered that biofilms are actually the natural state of cells, and it has been observed in multi contexts. For example, it has been found in clinical settings, in water supplies, as well as in the food industries. Past studies has been mainly focused on the monospecies biofilm formation, while growing efforts are towarding to the multi-species biofilm formation by reconstituting those multi-species isolates and then grow them under the simulated environment to understand their interactions. Another point is multi-species interactions also contribute to the persistence of microbes. For example, the non-pathogenic Pseudomonas putata and Pseudomonas fluorescens are the common food colonizers. They have demonstrated their ability to protect Listeria monocytogenes against the regular cleaning and disinfection routines. And then all of this, both biofilm formation as well as the multi-species interaction within the biofilm, they both contribute to the long-term persistence in the food processing facilities. So to study the microbial diversity and composition in the food processing facility, 16S amplicon sequencing has been used as a main standard to review the surface microbiome and the food product microbiome. Metagenomic sequencing recently are gaining more interest in food processing facilities because they can also provide the functional profiles. If we want to know whether those isolates are the same strain or not, post-field gel electrophoresis was the gold standard to for the outbreak analysis and track the food pathogens. However, when the next generation sequencing become more affordable, whole genome sequencing now is becoming the new standard to subtype the food pathogens. For example, Listeria monocytogenes and E. coli 0157. If we want to know whether those isolates are the same strains, typically we will use the single nucleotide polymorphism, which is the genetic variant in the DNA sequence to predict whether they are the same strain or not. The term of strain persistence can acquire many different meanings. In this talk, we define strain persistent or the persistent occur in the processing facility if the same strain or the same isolates, the different isolates of the same strains has been isolated multiple times at different location or different times in the same processing facilities. Here is an example of research conducted in a co-smoked salmon processing facility. What they did is they collect 40 whole genome sequencing data of Listeria monocytogenes from 1995 to 2015. Well, they found that those 14 isolates of Listeria monocytogenes are closely related, and then the strain has been harbored the same strain within that facility since 1995. Which demonstrate the long-term persistence of the Listeria monocytogenes and then pose the potential outbreaks for the Listeria monocytogenes. However, we don't have this much detailed data related to food spoilage microorganisms. So my project start with a fairly straightforward question: is there a persistent microbiome of the spoilage microbes? To answer that, I have been visiting this pork processing facility located in Alberta twice within 6 months. What I did is I have collected multiple environmental samples, including both food contact surfaces, non-food contact surfaces, and as well as meat samples. The meat samples were collected to map the meat product microbiome to the surface microbiome. The surface was also collected during production as well as after cleaning and sanitation to reveal any bacteria can survive after the sanitation routines. In total, I have collected 46 samples in September 2022 and 73 samples from March 2023. And then there are roughly 2,000 isolates has been collected. What I do is, once I have the sponge samples of the environmental surfaces or meat samples, I enumerate the sponge samples with some peptone water to encourage the growth of total aerobic bacteria, lactic acid bacteria, as well as Enterobacteriaceae. And then after getting the mixed colonies, what I do, I will do the subsequent streaking to get the single morphology of the microbes. And then from those 2,000 isolates, the clonal isolates of each sampling site were excluded by the rapid PCR. After that, roughly around 1,000 isolates has been sequenced by the nanopore whole genome sequencing platform. The subsequent analysis include genome assembly as well as species identification against the genomic taxonomy database. I also did a phylogenetic analysis and SNP analysis to infer whether those isolates are the same strain or not. In addition, in case I didn't isolate all the microbes in the sampling site, I also ran the 16S amplicon sequencing to review any missing microorganisms. Okay, so obviously there is a lot of data here, and I would love to sit and tell you everything about this, but I would like to save this when you read my manuscript. So overall, what I found is there is a very diverse microbiome in this processing facility. Across 4 different phyla, 47 genera, and a total of 137 species has been identified. In addition, there were 74 potential new species can now be classified based on the current taxonomy database. More important, there was no pathogens has been detected at both sampling time. High occurrence and diversity has occurred among Pseudomonas species. For example, those 3 Pseudomonas species has occurred over half or 1/3 of the sampling sites in both sampling times. After storage, the meat samples mainly consist of lactic acid bacteria such as Lactobacillus cuvatus, Carnobacterium divergens, and Carnobacterium maltoromacticum, as well as some Enterobacteriaceae, including Ranella, Serratia, and Haffadia. Strict aerobic bacteria such as Pseudomonas are eliminated after the 3 months storage. Most of the microbes can survive after the cleaning and sanitation. However, some genera such as the Acinetobacter, Xanthanobacterium, Saccharobacter, as well as Serratia were not covered or recovered from the sanitized surfaces. So remember, I also ran the 16S sequencing to review any isolates, reveal any microorganisms that we might miss by the culture-based approach. So this is how the data look like by the 16S sequencing. What we did is we compared this result with our culture-based result to reveal, to show what is there and what we have missed. So overall, from this figure, you can see overall over 2/3 of the microbial communities has been fully recovered by the culture-based approach. However, there were 6 samples, which has very tall stacked bars, has less than 25% of the microbial taxonomy being recovered by the culture-based. Those microbes mainly come from the dead cells because you can see here, a lot of them are from the sanitized surfaces. There are also some low biomass samples considered as a kit microbiome present. For example, the wall surfaces in the shipping truck, as well as the pipes in the facility. There are a few genera present low abundance. For example, the Delilioa and Xanthanobacterium has been constantly identified by the culture-independent approach, but then missing in the culture-based approach. What I want to highlight here is conversely, there were 41 species only detected by the culture-based, but either not detected or present underrepresented by the sequence-based approach. So further to know how those microbial communities overlap with each other, including the food contact surfaces, non-food contact surfaces, as well as the meat samples, we use the Bray-Curtis dissimilarity analysis to review the community dissimilarity. For example, we have done this analysis based on the 4 different classifications. The first one is a type of surface. So we group the surface, whether they are food contact surfaces, meat surfaces, or non-food contact surfaces. We also run the analysis based on the location because the sponge samples were collected from both cooler rooms, cutting floor, peeling floor, as well as the packaging area. We also compare the communities before and after sanitation, as well as using the zone concept to review is there any difference. So overall, what we found is that microbial community only differs before and after sanitation in panel C. So there was no difference on the microbial communities on both surface types, zones, or the locations. By using the network analysis, we can further to interpret our data to review their bacterial interactions on both environmental and meat surfaces microbiome. What we found is that all of the isolated microbes demonstrate positive correlation. That means there is a synergistic interactions among the microbes in this food processing facility. And then we also identified multiple clusters with the key species of genera Pseudomonas, Xanthanobacterium, Saccharobacter, Acinetobacter, and Pantonia at the center. Some spoilage associated microbes, such as Carnobacterium, as well as Lactobacillus, has been co-occurred with gram-negative microorganisms. And more importantly, as you can see in this figure, you probably see a lot of Pseudomonas species. They has been correlated with other Pseudomonas species or other genus. That means they play very important synergetic roles for the microbial profiles in this processing facility. So to review the dispersal and persistence of the Carnobacter mottle, the persistence of the spoilage microorganisms, what we do is we focus on those 5 different species which has been isolated from the store meat samples. What we did is we run the phylogenetic tree of each target species. For example, this is a phylogenetic tree of the Carnobacter mottle macticum. Different colors represents the different sampling sites or the different sampling time. As you can see, there are a lot of isolates of the Carnobacter mottle macticum has been isolated from both meat samples, from the food contact surfaces such as brick table, as well as the non-food contact surfaces like the floor drains in the cooler room. The isolates has been also collected at both sampling times. And if you take a look at the bottom this cluster, you can see that a lot of quite many meat isolates are closely related to the non-food contact surfaces, especially the floor drains at the cooler room. So to further know whether those isolates are the same strains, what we did is we run the SNP analysis. On a side note, so the current FDA SNP calling pipeline is mainly used for the food pathogens as well as for the short reads generated by the Illumina. As our sequencing was conducted based on the nanopore platform, the the FDA pipeline is now actually suitable for the SNP analysis. So our lab has been develop a new workflow with the use of the CLAR3 to run the pairwise SNP analysis. So this figure represents the repeated isolation of those 5 targeted spoilage-related species across the processing facility. And we used a stricter threshold to determine whether they are the same strains or not. So FDA used 20 SNPs and we used 10 SNPs. Different symbols represent the different species. For example, the circle represents the Carnobacterium multimacticum, while the square represents the Carnobacterium divergens. And then if the symbols are color-coded with the same color, that means they are the same strains. So overall, what we found is all targeted species or strains were isolated at both sampling times. The microbes can disperse across the fabrication rooms as well as the cooler rooms. If you take a look of this killing floor, there's no targeted species has been recovered in this killing floor. Also, we also found the microbes can persist over 5 months. This is simply because the microbes present in the first sampling time also has been detected at the second sampling time. And then if you take a look of the Carnobacterium, if most of the Carnobacterium multimacticum from the meat isolates can be traced back to the cooling floor, while another hotspot for the persistence of microbes is the packaging area. So to further know what are the driving factors actually plays a role for the persistence of those spoilage microbes, we reconstitute our bacterial communities to let them to grow for biofilms. What we did is we picked 10 sampling sites that including those spoilage related microbes, including Carnobacterium and Serratia species. We put them together, we let them grow on the full grade stainless steel surfaces, and then grow them at both ambient temperature and the 4-degree temperature to mock the simulated processing environment. What we found is that actually the reconstituted microbial communities formed denser biofilms at 4-degree than the 25-degree. And if we analyze the microbial profiles in those reconstituted biofilm communities, what they found is that both Serratia and Carnobacterium can resident in the biofilm communities and coexist with many other different species, for particularly the Pseudomonas species. So, so now what I have shown that who has been there in this processing facility and where is the problem. For example, the cooler drains and the packaging material. For the next talk, I will go through to show our findings on the ozone nanobubble solution as an alternative intervention strategies to control the meat spoilage. I will present our findings based on both lab results and the in situ results. So ozone in the last decade, ozone actually has becoming more popular because there's a huge demand for the consumers for the greener sanitizers. It has been considered as safe to use and has been used in many different food types. For example, washing of the fruit and the vegetables, as well as sanitizing the equipment surfaces. The main problem is ozone is very unstable, especially when it dissolved in water. So due to the instability of the ozones, they can rapidly deplete to oxygen and compromise their sanitation efficacies. However, these limitations can be made or solved by the introduction of nanobubbles. So if you look at this figure here, we have the traditional macro and micro bubbles, which are a lot larger compared to the nanobubbles. Nanobubbles is the nano-size cavities that has demonstrated longer duration in the liquid solution compared to the micro and macro bubbles. So if we think about the aqueous ozone has been applied in the food processing facility, this figure shows like the aqueous ozone is made up with both micro and macro bubbles, which are irregularly shaped, and they can now cover the entire surface of the equipment surfaces. However, if we combine the ozone nanobubbles entrapped with ozone entrapped with nanobubbles, they can uniformly distributed across all the equipment surfaces and thus improve the sanitation efficacies. So what we did is by the help with Solution, a startup company from US, Austin, they help us install 2 ozone nanobubble systems in both our labs as well as in the meat processing facilities in Alberta. In our lab, what we did is we inoculate strain cocktails, including the spoilage microbes, for example, the Broxol, thermosurfacta, Carnobacter maltoromacticum, Leuconoxoc, Galidium, Lactobacillus sakei, as well as some Enterobacter ca like Yersinia and Hafania. What we did is we collect some meat samples, we sterile them, and we inoculate our spoilage cocktails into the meat samples. And then after that, the inoculated meat samples were treated with either ozone nanobubble with water as control or with the peroxide acid. And then after the treatment, the meat samples were stored in the 1-degree refrigerator, and then we collect the samples at different weekly intervals, and then compare the microbiota in the stored meat samples. In the food processing facility, we have 2 different systems installed. One is installed as water hose, mainly to wash the offal buckets as well as split saws or razor knives in the killing floor. Another system is a low pressure spray, mainly used for the pork carcass, and then to spray them and then to see their antimicrobial effects. After the spraying for the meat samples, I also collected some meat samples and run the microbial analysis to review the microbial profiles before and after ozone nanobubble treatment. So this is how the ozone nanobubble equipment look like. This is in our lab. As you can see, we have an oxygen cylinder. The red box is the ozone generator. And then we have different holes connected. And then this tank is full of ozone nanobubbles. So based on the result, what you can see here that the black circles represent the untreated samples, which are inoculated, but there's no treatment. The gray circles represent the meat sample that has been washed with water as a control. And then for the blue squares represent the inoculated meat samples has been treated with the ozone nanobubbles. And then the orange diamond represents the meat samples washed with peroxide acid. And then different panels represent different strains. For example, A is the Broxothorax, B is the Lactobacillus, and C is the Yersinia, D is the Carnobacterium, E is the Leuconoxoc, and F is the Hafania. What you can see here is that the ozone nanobubble plays very minimal or slight disinfection against all species or strains, except for the Yersinia strains. For the peroxide acid, it does also demonstrate very minimal effect bactericidal effect against those gram-positive bacteria such as lactobacillus as well as leuconoxoc. However, they have relatively or moderate bactericidal effect against the gram-positive bacteria such as Hafania and Yersinia. So for the in situ validations, what we did is we collect 10 different offal buckets, air knives, and as well the split cells, and then wash them with either water or with the ozone nanobubbles. What you can find here is for the offal buckets, ozone treatment on these bucket surfaces can achieve 1 log, at least 1 log cell reduction compared to the untreated samples. For the air knives, there is also a significant difference compared to the treated samples and the control samples. However, for the split cell samples, we didn't observe any difference. This could be due to the sampling problems. To further review to map the ozone nanobubble application in the food processing facility, we also choose 10 carcass that has been washed with ozone nanobubble under the low pressure spray. And then by having the microbial results of the carcass washed by the ozone nanobubbles, we compared this result to the historical result, which has been mainly used for the peroxide acid wash in the processing facility. What we found is that constantly there's around 1,000 CFU per centimeter squares on the pork carcass. After peroxide acid wash, this microbial loads can usually be reduced to 10 CFU, so 10 cells. However, in our 10 ozone nanobubble carcass, we did find that this microbial loads can be further reduced down to 1 cell, which demonstrates that ozone nanobubble actually has been working effectively on the carcass in the food processing facilities. To further review the effect of the ozone nanobubbles on the microbial profiles in the meat samples, what I did is I also collect some meat samples from the production line, including the pork legs and as well as tenderloins. I take 2 legs and 2 tenderloins. One of each was sprayed with water, another one is sprayed with ozone nanobubbles. And then I let them store at their processing facilities for a period of 3 months. And after that, I will collect the samples and then profile what the microbial communities or these diversities in the stored meat samples. Overall, there was no color change or off odor generated after the ozone nanobubble wash. And more importantly, we found the microbial diversities did vary between and after the ozone nanobubble treatment. As you can see here, certain microbes such as Clostridium and Vigrococcus in this dark blue bars are significantly reduced after the ozone nanobubble treatment or completely removed after the ozone nanobubble treatment compared to the control groups. The different meat samples also have different microbial profiles. For example, in the leg samples, Morganella as well as Clostridium and Leuconoxoc was only observed in the leg samples, but absent in the tenderloin samples. Serratia, Carnobacterium, and Yersinia has been detected among all 4 meat samples. And then the Serratia species particularly has increased after the ozone sanitation compared to the control groups. Another thing I want to highlight is that for the Clostridium species, this has been fully eliminated or below detection after the ozone treatment, which we hypothesized that when the ozone nanobubbles in contact with the meat samples, the ozone depletes into oxygen, and then the presence of the increased level of oxygens might inhibit the growth of the Clostridium. That's why there is not present in our treated samples. So overall, this study has identified a large and diverse microbiome within the food processing facility. They are able to disperse across different sampling sites. That means dispersal spatially as well as persistent over time. In our work, we did find a persistent over 6 months. The ability of those microbes either form biofilms or coexist with each other has been further contributed to their dispersal and long-term persistence within the food processing facility. The application of this ozone nanobubble actually can be considered as a novel sanitizer and demonstrate potential use to control the microbial load and also alter some microbial diversities. So by doing these two studies, now we can ask and answer many questions. For example, which sites requires specific cleaning? In our study, we found the cooler drains as well as the packaging area, including both food contact surfaces and non-food contact surfaces, are the hotspots for the contamination transmission. By having these large isolates, we can also further ask and answer many questions, including how does those microbes interact, how do they communicate with each other, or what communities actually can work as the background microbiota and accommodate the persistence of other food pathogens. So to find those answers, I would like you to read my thesis. So it is almost public available. I defended last month, and then it will be available, and I look forward you read about it. So with that, I want to specifically thank the Canadian Institute of Food Science and Technology for providing me this opportunity to share my PhD studies with everyone. I also want to, of course, thank all the funding agencies to make these studies available. I also want to especially thank my supervisors, including Dr. Michael Gänzle and Dr. Xianjing Yang, for their valuable support and guidance throughout these studies, as well as the collaborators such as Fee and Yannick for their help for this research. With that, I'm happy to take any questions.
Okay, thank you very much, Shaelyn. You did a wonderful presentation. Okay, with this, I'd like to, on behalf of the CFST Food Cluster, thanks to Shaelyn Xu for the presentation, and also to make an announcement here. Our next webinar is on October 30th at noon. The topic will be plant protein phenolic complexation towards healthy plant-based food development. The speaker will be also from University of Alberta, Professor Lingyun Chen. Well, with that, if there's no further questions, I'd like to join the meeting, seminar, and look forward to see you all on October the 30th. I appreciate your active participation to this webinar and look forward to have your input for the next time. Thank you. Bye for now.